Blue in what node?
Okay, I'm going to speak, as Dan mentioned, about RNA editing. So, what is RNA editing? We know that the information in the genome, we believe, should be identity and kept through all over the process of genetic information processing in the cell. The RNA should be identical for the DNA, where it's transcribed from, and the proteins that emerge should be translated based on the genetic table that we know. However, there are two protein families that not obey this very common knowledge rule. And I'm going to speak about ADAR, and ADAR is changing adenosine by deamination into inosine in the RNA. So you have A in the genome, specific position, but if the other enzyme can, you can change the A in the RNA, specific position into I, and I is already a G. So you actually have a change from A into G at the RNA level, and later on, if you look at the genetic table, it's manifested also in different proteins. And also, there are other protein families that we're now working very hard on it, but I'm not going to talk today at all, are C2U protein families that change C, also by determination, into U, and add another level of complication for the flow of genetic information. So the other proteins, there are three in India, but they are occurring all over it as well. And we have in human other 1, 2, and 3. All of them have the aminase domain, where they do the deamination, and all of them have double-strand RNA binding. So they bind to double-strand structure in the RNA, and this is obligatory in order to do the editing. So you need specific structure in the RNA in order to recruit the ADAR. It's important. Without ADAR, none ADAR 1 or ADAR 2, you cannot live. It's embryonically or shortly after birth, it's lethal. And it usually takes place before splicing. So the double-strand structure can take place in the pre-mRNA, not necessarily in the mRNA. And editing is expected generate diversity in our transcriptome and also in the proteome. So you can see in specific position alteration that can be combinatorial. Unlike alterations derived from mutation in the genome that should be an haplotype, that are linkage between all of them, you can have, let's say you have genes that have 10 sites, you have 2 to the power of 10 different isoforms that can take place in different cells, different time, in the same cell in different time, in different tissue, different person. So it adds a huge level of diversity to the transcriptome and also in some cases in the proteome. And I mentioned that it's usually in the transcriptome, not in the proteome, because almost all editing takes place in non-coding region, at least in human. In alu repeats, you have alu repeats in our genome, but we have a million copies of alu, slightly more than a million. And because we have so many of them, if you have in the pre-mRNA of a gene 2 alus in reverse orientation, they can form, before the splicing, this double-strand structure that eventually is going to be edited. And we know this for many, many years, actually. I work on this for 2 decades. And we know that there is extremely abundant, there are a million copies of alu in the human genome. All of them, virtually all of them are related to some degree. So we speak about tens of millions of editing sites that take place in the human transcriptome. But I want to remind you that alu is primate specific. So all this party of editing is in alu is something that takes place only in primate. And we also, and I have to mention that majority of this work done with Eli Eisenberg from Tel Aviv University, that we are working on this again 2 decades now. So we can now look at in the genome, and simply by looking for what's the probability of an alu to form double-strand structure. I mean, what is the distance from the nearest alu in reverse orientation? How similar are these 2 alus to each other and can form stable double-strand RNA? Tell us what is the editability of specific alu. So simply by looking at the human genome, we can tell which alu is going to be highly edited. But editing is not limited for human. When we did editing analysis across many, many organisms, we find that editing actually is common. Also, alu is unique to primate. We see editing everywhere, in every metazoa that we look, starting from Cora, very, very far in evolution. And actually, human is not one of the highest edited organisms. Interestingly, most of the animal models that we work in, mouse, C. elegans, in fly actually are outliers with the relatively low level of editing. So, utilizing the same idea of understanding the genome architecture in humans, the human genome that allows us to understand where editing takes place, we look in the genome of all these organisms, and actually you can understand why there are organisms with high editing and some organisms with low editing. Humans, as I mentioned, have one dominant sign, one element that we have a billion copies of it, so which easily can form double-strand structure. Mouse, on the other hand, that have low editing level, have 4 different signs. Each of them is very short, less than 150 base pair, I mean 300 base pair long, and much more divergent. So the probability to form stable double-strand structure is much lower. Therefore, editing is very low in mouse. In xenophus, for example, in the other hand, have one element, Erdinger, that is polydrome. So it can form double-strand structure without the need to have the similar family member nearby. So it's common, it has a lot of editing in this element. But as you can see, one outlier is the octopus. And in the octopus, we see a huge amount of editing, not only in the non-coding region, not only in the mobile element, but also in the protein sequences. And virtually every protein in the cephalopod lineage, octopus, squid, and scibia, we see extensive editing in the protein. For example, in alpha-spectrin, the genes that have more than 100-something editing recording event, you have 2 to the power of 100 different isoforms in the transcriptome. And it's not only theoretical number. We extensively sequence several genes in the octopus and squid in the depth length of almost a million. And we didn't reach to saturation of the number of different isoforms. So editing is really combinatorial there, and we didn't see one dominant variant. So when we also look for proteomic data, also the proteomic data obviously is very low level and represent only small portion of the data, we still see a huge amount of editing everywhere. So when we look, and I forget to mention, this is correct only in the neuronal tissue of cephalopod. So when we look at the cephalopod, we see, we have to understand that you have a huge amount of very different and huge variety of different proteins in the same animal, unparalleled diversity in protein sequences, and we don't understand why it is. I want to remind you that cephalopods are much more smarter than all others, mulassacas, that don't have the intelligence, so maybe supporting for this idea is that editing is located only in the nervous system. Maybe this diversity contribute to their brain complexity. Interestingly enough, when we look in the cephalopod, there is one outlier that is more earlier in evolution, it's the nautilus that don't have this composition. And another distinction between these two portions of the cephalopod lineage is that the octopus and squid live very short-lived. And we presume, and it is only a speculation, but also we have some initial events, indications that maybe the cephalopod have enjoy from this diversity of protein, but if you have so many proteins, many of them simply do not fold properly due to the, and we suggest that they may die simply because of this malfunction, accumulation of misfolded protein. And interestingly enough, the nautilus that don't have this editing live way more, can live decades, while octopus can be huge, but still will live 2 to 3 years. So this is another interesting outliers. But so editing is everywhere. But why? So maybe cephalopod utilize it for protein diversity, but why we see editing mainly in mobile element? What its help? And this question was addressed by several groups in the last decade. And editing serves as a defense mechanism against false activation of the immune system. For a cell, the most dangerous phase that you can think about it is to be infected by viruses. And many viruses have double-strand RNA structure. So one of the guardians of our cells, and this is true for every cell in metazoa, there are many guardians again that have to alert from viral infection. So one of them is protein called MDA5 that once you recognize double-strand RNA, it's for a filament across them, and this filament activates the immune system, mainly the interferon pathway, to the MAVS pathway. And this is very dangerous if it's derived from endogenous double-strand RNA. We understand that double-strand RNA is hallmark of many viruses, but as we understand, many RNA in our genome have this double-strand RNA structure, and therefore editing already in the nucleus, while you recognize endogenous double-strand RNA, edit it, and by that avoid the false activation of the MAD5 recognition, and this is to recognize the self against the viruses. And indeed, while other knockout is lethal, other knockout together with MDA5 knockout can live happily. So this is something that we understand. It's very important for the elimination of false activation of the immune system. So now we understand it, we develop computational tool to calculate what is the editing level per tissue. So once we understand that editing is important for immune activation, we can look for RNA-seq data and determine what is the editing level in this tissue. But in order to determine this is not a trivial task. If, for example, we simply count the mismatches that can be hallmark for editing, it depends on the coverage, on the rinse length, and all the methodologies that were available to calculate what is the editing level per tissue were not worked properly. So we developed the RNA editing index, which allow us now to take an RNA-seq and simply give a number after many normalization of what is the editing level of specific RNA-seq data. So now we can utilize and take every RNA-seq data and give it a grade. What are the editing level in this RNA-seq? And compare. We can compare various tissues, disease, conditions, and because we understand that, and obviously we can utilize it, we can define the regulator of other, but now we can utilize the publicly available data and look for places where editing can play a role. And first work was in autoimmune disease that we understand that some of them can be related for editing, because editing, now we understand, if you have less editing, you have more double-strand RNA, more activation in the immune system. This is exactly what we see in atopic dermatitis and psoriasis. We have less editing, which means that we have more double-strand RNA that can contribute to the vicious cycle of the immune activation. So this was a correlation. We find that there is a correlation between low editing level and activation of immune activity, but we want to go further to see if there is colonization. So if lack of editing can lead for autoimmune disease, and together with Yuval Dor, they create a mouse without editing activity in the beta cells, and this mice develop type 1 diabetes. And this actually was very exciting in the field, because it was very difficult to create model for type 1 diabetes, and here, simply by blocking the activation of the adder, you get all the phenotype of type 1 diabetes, and this, again, very exciting. Now there are many follow-up work on this. And also in cancer, we know that the immune system involved in fighting against cancer, and what we see across all the TCGA, that there are more editing in cancer tissue compared to the matched normal, and this is very exciting because we know now that if you can recruit the immune system, checkpoint inhibitors and all the other immunoregulator, we can activate presumably to help the cancer phenotype. And indeed, together with several groups in Boston, we managed to show that when you eliminate ADAR, you can have more activation or activity of the checkpoint inhibitor. And this is now there are several startup companies, and not only startups, that now developing other inhibitors exactly for this aim. So this was the first part of the story, but I want to, okay, so, but editing is not only localizing mobile element, but also in coding sequence. It can generate diversity in protein sequences, and it was difficult to find where editing takes place. Basically, it should be straightforward. You should simply look for RNA-seq data differences between the RNA and the DNA and look for mismatches. but these mismatches derive from sequence error, mutation, and many other reasons. So it was not easy. And to detect editing in ALU is relatively easy because we have many editing in the same position, so it's easy to find the mobile element, the cluster one. But the position was very difficult. And we know this is something that we worked 10 years ago, even more, that we find several evolutionary conserved sites. It took almost a decade to decipher what was the function. In this case, the FLNA, we find that this specific editing site in this gene FLNA is very important for the proper phenotype of cardiovascular system. But there was no atlas for where editing takes place in the coding. And just recently, we published an atlas of, again, with Eli, of all the editing sites in coding sequence. There are 1,500 such sites that are taking place in our bodies that change. So there are millions of sites in the mobile element and dozens of hundreds in the coding sequence. I will not go into much details how we did it. It's technically published. So, but just to say that it takes place in virtually every tissue with brain, but also cardiovascular system as the main system. We can tell you something about the. I have four. Really? Okay, so I skip. Okay, this is. So I skip together for another.
Unless you don't want questions.
Okay, I will skip for a completely different story. That how to utilize RNA editing in order to fix mutation. We're all aware about the CRISPR and other base editor technologies that allow us to fix mutations in the human genome, but all that suffer from very severe issues. One of them is that you have to introduce a foreign protein from outside, the Cas protein, and they can off target. So what we did in the last few years is an effort to utilize endogenous ADAR to recruit him to change mutation by simply introducing the double-strand structures that have it, that is needed. So together with Shragi, we find some of the key features that ADAR needed, all in the process to find what are going to be the perfect guide RNAs. We will put the ADAR, and we find some features that are important, but together with Shai, we develop a yeast system that allows us to screen hundreds of millions of different combinations to find what are the oligos that we need in order to recruit the ADAR to go into a specific position. And this is an example of one effort. When we have mutations that we want to fix, we get from the many oligos that we can, some of them can generate editing. It can fix many of the genetic diseases, not only the ones of ATG. Many of the cancer-derived diseases are potentially targeted by ADAR. And I didn't mention it, but also in just 2 weeks ago, our paper led by Stefan Storten from Tübingen that was we're allowed to fix Rett syndrome already in live animal, of mutation of model of Rett syndrome. So I have to summarize that there are millions of sites in ALU. We find several hundred coding. It's abundant in metazoa. Alternation editing can contribute for productivity in cancer. Other inhibitor are possibly for anti-cancer drugs. And other is a new promising tool for endogenous base editing of clone mutation. And with that, I want to thank. And I really sure think that you owe me 3 more minutes. Thank you very much.
Question?
To what extent the level of RNA editing in coding regions varies among individuals within the same population, the same species, the same tissue, and whether it is fixed or not?
Okay, so when you look for the RNA editing targets, you can divide this into 2 classes. One of them are the sites that evolution worked very hard on them. So which means there are selections that the editing is important. And in this site, you see very tight regulations. The editing level among the population is very, very similar. And we look for all the genetics data. And this is true to the ones, and actually they are going to be changed in different tissue. But in the same tissue, across many patients, people are about the same. But this is only the minority of sites. Majority of sites looks to us that are simply off target, in positions that they don't really care about, in positions that change nothing. And in this, you can see large diversity, and this is the majority of sites.
How come that the same immediately is not active on intruders like viruses?
In viruses?
You said that other is used to protect our viruses, but your viruses attacks also.
Exactly. So viruses, as we know, viruses are very. They are not modified? So viruses are very, very smart. So we know about one virus, Delta virus, that is known that it's going to be edited. So already come with a stop codon that only after the editing is going to be active. So viruses learn also how to utilize it, but it's unsolved issues still in under research is if other is only mark the endogenous double-stranded RNA that will not go to avoid false activation, but also what is how important editing is against viruses, because many of us we know from several papers that COVID-19, for example, undergo editing, but we don't know for sure if it's beneficial to the viruses, it's act against the viruses, so it's on open questions.
So I wonder, you said that at least in humans, RNA editing occurs a lot in the allo. Okay. So as we all know, in cancer and there are other types of diseases where genome instability occurs. So in genome instability, part of the genome instability is the instability of the repetitive elements, and I wonder whether this can influence to some degree the levels of editing.
Okay, so when people refers to misactivation of a mobile element, usually they refer to the mobile element avoid their methylation and start to express. So when this takes place, you have one allele that is expressed. It's not formed double-stranded RNA. It's one element. In order to have RNA editing, you need two elements in the same transcript in order to form this double-strand structure.
Head to tail.
Head to tail, but in the same. Both, but in the same, yes. But in the, if you have activation of one of them, it's not something that provoke editing. However, as you mentioned, you have genomic alteration, and you can think about conditions that can form now the outcome will be double-strand structure. You can think about regions that have double-strand structure, but typically are methylated, and now they are unmethylated. And yes, this is something that I think it's a great question because we're actually trying to think if we can see selection against these events in cancer. But this is, again, a question and a good one.
One other question that I had in mind, there's the growing field of transcripts coming from regulatory elements such as enhancer transcripts, and I wonder whether such transcripts also undergo editing, and in this way, you know.
So we don't know about all type of RNA regulation, but one very common, very famous, is microRNA. MicroRNA in the early stages, in the pre-microRNA, have long double-strand structure. And this is subject to editing as well. So many microRNA, not many, some dozen of them, have editing in the microRNA. So you can think about if you have different versions of the microRNA, because of the editing, you have completely different set of targets if it's in the seed region. So another level of complexity, which again, very exciting. And the last one is circular RNA. Circular RNA, in order to form a circular RNA, need double-strand structure to form a splicing. This is also edited. So we together we managed to show that editing is reverse correlated with accumulation of double-strand structure of circular RNA.
Any more questions? Let's thank you.